Hello everyone, today it is a great honor to introduce our research. Inhibition of ferroptosis by microbiota-derived lithocholic acid underlies the intestinal radioprotection of a host defensin-derived oligopeptide. Please refer to the graphical abstract of this study. Human defensin-5-derived oligopeptide 89-CG enriches the intestinal bifidobacterium pseudolongum and increases the lithocholic acid LCA levels. In cell, mouse, and human small intestinal organelle models, LCA demonstrates radioprotective effects. Mechanistically, LCA activates TGR5 and upregulates SREB-POM, which transcriptionally regulates the SCDAN expression and remodels lipid metabolism, thereby inhibiting ionizing radiation IR-induced ferroptosis. The highlights of this study include: first, the human defensin-5-derived oligopeptide 89-CG alleviates IR-induced intestinal injury IRI. Second, 89-CG enriches intestinal bifidobacterium pseudolongum and increases LCA levels. Third, LCA upregulates SCDAN through the TGR5 receptor and its IR-induced ferroptosis by reprogramming lipid metabolism. Result 1. The H5-derived oligopeptide 89-CG effectively alleviates IRI. Using a total abdominal irradiation mouse model for drug screening, we found that the H5-derived oligopeptide 89-CG oligopeptide mitigates IR-induced intestinal mucosal damage and improves mouse survival rate. Result 2. 89-CG alleviates IRI by reshaping the gut microbiota. Fecal microbiota transplantation experiment revealed that the gut microbiota mediates the radioprotective effect of 89-CG. Metagenomic sequencing analysis demonstrated that 89-CG increases the abundance of the probiotic bifidobacterium pseudolongum. In vitro experiments confirmed that 89-CG can promote the growth of bifidobacterium pseudolongum. Result 3. 89-CG increases the radioprotective LCA in the gut. Metabolomics studies revealed that 89-CG increases intestinal LCA levels. In multiple models including cells, mice, and human small intestinal organelles, LCA exhibited inspiring radioprotective effects. Correlations indicated that the increase in LCA is associated with the enrichment of bifidobacterium pseudolongum. Single bacterium transplantation confirmed that supplementing with this probiotic elevates intestinal LCA levels. Result 4. LCA reduces IR-induced cell death by upregulating SCD1 expression to inhibit ferroptosis. Proteomic sequencing analysis revealed that in irradiated cells treated with LCA, the ferroptosis signaling pathway is significantly enriched. By improving mitochondrial morphology, reducing lipid peroxidase, and decreasing intracellular iron accumulation, LCA was demonstrated to counteract IR-induced ferroptosis. Further lipidomic studies revealed that LCA remodels the lipid metabolism in irradiated cells. Based on interpro enrichment analysis, we found that LCA upregulates SCDAN expression in irradiated cells. Gene silencing assays confirmed that SCDAN knockdown attenuates the abilities of LCA to reprogram lipid metabolism, suppress IR-induced ferroptosis. Result 5. LCA upregulates SCDAN expression by activating TGR5 SREB-POM signaling in vitro and in vivo. Gene silencing screening revealed that LCA-induced SCDAN upregulation depends on the membrane receptor TGR5. LCA activated TGR5 to promote the expression of the downstream transcriptional regulator SREB-POM, which directly binds to the promoter region of the SCDAN to enhance its expression. Result 6. SCDAN inhibition or barrel knockout diminishes the radioprotective effect of 89-CG in mice. Transcriptomic analysis revealed significant enrichment of the lipid metabolic process in irradiated mice after 89-CG treatment, with SCDAN being notably upregulated. Administration of the SCDAN inhibitor MF438 attenuated the radioprotective effect of 89-CG. Additionally, barrel knockout impaired the efficacies of 89-CG to induce SCDAN expression and alleviate IRI. Our study revealed that the human defensin-derived oligopeptide 89-CG alleviates IRI by enriching bifidobacterium pseudolongum in the gut and elevating LCA levels. Mechanistically, LCA upregulates SCDAN through activation of the TGR5 receptor signaling pathway. This promotes monounsaturated fatty acid and reprograms lipid metabolism in irradiated cells, ultimately suppressing IR-induced ferroptosis.